Okay, so welcome to the webinar with Ruchi Agashe, who is our speaker today. So thank you so much, Ruchi. And this event is hosted by the Helix Initiative. So I'll do a really quick intro of Helix. So Helix is the world's largest youth research community with around, I think, 2,900 members now. And members can participate in STEM leadership, research, volunteering, and competition opportunities for free. And we also do have volunteer hours available. So make sure to join our Discord via helixscience.org, and I can put that in chat later on. You can start a chapter, you can do tutoring. We also host hackathons. We also have a research program, which we will be opening sometime soon. And we also have a social media account on Instagram, Twitter, YouTube. And we also host seminars like these. And you can sign up for the other science nights here, and I will also be putting this one in the chat. I think this is our 10th science night of this series, so that's pretty cool. And now it's time to meet our amazing speaker, Ruchi, and I think Sid will take it from here.
Yeah, thanks for introducing Helix, Sarah. So it's my honor to present our speaker, Ruchi Agashe. Ruchi is an incoming sophomore at Yale University, majoring in molecular, cellular, and developmental biology on the premed track. She's currently doing research at the Yale School of Medicine and interns for Planned Parenthood in Southern New England. She's on the board of Yale Undergraduate Research Association, YURA, period at Yale, and MedSci. She is also the Community Health Educator on campus. After completing her undergraduate studies, Ruchi aspires to attend medical school and become an OB-GYN. So without further ado, I'd like to introduce our speaker, Ruchi Agashe.
Hi, everyone. Thank you guys so much for showing up. Sid already introduced me, but yeah, my name is Ruchi. I'm an incoming sophomore at Yale. I did a lot of research in high school, and so it was really exciting to be asked to speak about my research and talk to everyone here. And so I had a slideshow I prepared just to talk about my research, and then I'll describe like what I did, and then at the end probably there'll be time for questions. So yeah, let me just share my screen. Okay, so my research, my project was titled The Identification of Treatments for Hemophilic Joint Disease through the evaluation of vascular defects via optimization of imaging techniques. I worked at the Scripps Research Institute under Laurent Mosnier in the Mosnier lab. So what is hemophilia? Hemophilia is a factor VIII deficiency. Factor VIII is a blood clotting factor. So hemophilia patients have trouble with blood clotting after an injury. And it largely affects males. And one major problem with hemophilia is this excessive joint bleeding. So hemophilia patients have excessive joint bleeding in weight-bearing joints, typically the knees. And this often leads to synovial proliferation and neovascularization, so changes in the vessels in these joints. And so having an injury, after injury, you have vascular remodeling. So the vessels start to grow back, and the vessels that grow back are larger and more fragile. And because of these more fragile vessels, you're more prone to joint bleeding. And you end up in this vicious cycle where you're just more likely to get spontaneous joint bleeds. And hemophilia patients often have a pretty low quality of life because of this. A lot of them can't walk. And just to explain the extent to which joint bleeding occurs, even stepping off the curb the wrong way can initiate a joint bleed. So it's not something that you can really prevent. The bleeds are quite spontaneous. And this more excessive form of hemophilia is called hemophilic arthropathy, which I'll probably be referring to as HA, separate from hemophilia A. So whenever I say HA, I'll be referring to this more aggressive form of hemophilia with joint bleeds. So my project goals. So I wanted to focus on this vasculature. Current treatments do clotting factor replacement treatments while they treat parts of hemophilia, they don't account for these joint bleeds. So people that are under factor VIII replacement or other clotting factor replacements, they still experience joint bleeding. And so my goal was to determine whether the vessels in hemophilia patients are abnormal and excessive. And one paper published before I had started my research had started investigating this. They basically saw that there was excess vasculature, but because of lack of techniques to look at the 3D visualization of the joints, it was harder to determine the actual shapes of the vessels. So I first wanted to just confirm the abnormal vessel formation. And afterwards, I wanted to know if this abnormal vasculature, which I had then at that point confirmed existed, I wanted to know if it was unique to hemophilia, or if it was seen in other joint diseases. Finally, next I wanted to look at whether or not the absence of factor VIII, which is the clotting factor, if that's what drives the abnormal vasculature. Then I wanted to know what angiogenic markers were expressed in factor VIII knockout models and could potentially serve as treatment targets. And finally, I looked at one promising treatment target and determined the effects. So this is a lot, but I'll break it down, starting with the first project goal, which was determining whether or not the blood vessels in hemophilia patients are abnormal and excessive. And so the way that I determined this was through 3D imaging, and it was actually quite difficult because like the current technologies when I was doing this project were very limited. There was immunofluorescent staining, which is basically where you take the tissues and you're able to stain for different factors, and that's what the paper before me had done. But the extent to which the information that you can get from that is very limited. They were looking at the CD31 and alpha SMA, which is like the outside of the vessels, but that information can only give you the amount of vessels and vessel diameter, but you can't actually visualize the vessels. So I wanted to create a form of 3D imaging and utilize 3D imaging to really look at the vessels. And the main problem with this was the fact that I was using mice who have even smaller vessels than humans. And I was looking specifically in the knee joints, so like really small vessels in mice. And so I first decided to use this technique called perfusion, which is basically where you fill the vessels with a solution. And so this was my first try, and it kind of failed basically because, as you can see, there's just one vessel and it's a really big vessel. And what I did, just to explain, is I filled the vessels with a solution that had barium sulfate. And barium sulfate is detectable under a micro CT scanner. So I filled the vessels with a barium sulfate solution and then visualized it under a micro CT. And I quickly realized that that's not a good idea. Like, it didn't work because you only have you can only visualize the bigger vessels. And I realized that I needed to optimize the solution that met 3 conditions. One, it needed to, be able to be visualized under a micro CT scanner, so it had to be barium sulfate based. 2, it had to be able to be less viscous so that it can actually perfuse the smaller vessels in the joints. And finally, it had to be able to harden within 24 hours so that I could quickly image it without any changes to the vasculature. So I spent a lot of time, a few months actually, literally just changing the ratios of barium sulfate, dilutants, to the point where I was able to get a solution that I could actually see the vasculature and I can actually see these smaller vessels. And this is an imaging I really like just because I think it looks cool. But this is the first ever 3D visualization of mouse joint vessels. Very cool, not just for my project, I think, but some of the labs I was working with, the lab that I was using their micro CT scanner, they're currently using this technology to visualize vessels in the spleen. It can also be used in the heart, in the brain. It's something that's very, versatile and can be used in a lot of other projects. So I think it was cool that something that I had optimized is now being used for other projects. But, for the purposes of this project, I was able to determine that the vasculature is abnormal in hemophilia. It's something that I was able to confirm the findings of the last paper. And after this, I wanted to ask, is this abnormal vasculature, is this specific to hemophilia, or do we see this in other joint diseases? And the main reason I wanted to know was because it was something that was very unique and I hadn't heard of before, but a lot of joint diseases that affect weight-bearing joints like rheumatoid arthritis or osteoarthritis, maybe some of the treatments for that are already accounting for these changes in vasculature, and some of these treatments could potentially be looked at for hemophilia. And so what I did was I actually got permission to use human samples from a hospital, which was really cool. And I was comparing normal patients, those with hemophilic arthropathy, rheumatoid arthritis, and osteoarthritis. And so these green circles here, these are blood vessels. If you can try to visualize that, you're taking like a section of it, and the outside of the blood vessels and the shape of the blood vessels can be seen through staining. And so in normal joints, you can see you have very circular, very small normal vessels. And then you look at hemophilic arthropathy and you have these massive, really weirdly shaped vessels. I think this one looks really weird right here. Like the fact that your vessels can look so abnormal and so large, and these are to the same scale. So this just shows the extent to which hemophilic arthropathy has abnormal blood vessels. And again, this reaffirms the last findings with the 3D imaging. When we look at rheumatoid arthritis, we see larger vessels, as you can see here and here, but it's definitely not, the shape is not to the extent of which you see in HA. You still have, regularly shaped vessels. And then osteoarthritis is just, very similar to the normal vessel formation. And I also quantified this, and what you can see is like when you look at vessel diameter only over 40 micrometers, you see there's like significant differences in vessel diameter for HA, but not really for rheumatoid or osteoarthritis. And the same has to go with the lumen thickness, and lumen is the outside of the blood vessel. So you do see some increases, but it's really not, significant enough. And even through here, you can tell that, it's really, this phenotype is very unique to HA. You don't really see it in other joint diseases. So it was a little bit disappointing. I thought it would be cool if other joint diseases had the same phenotype. It would definitely be something that would be interesting to look into. But it was also interesting to know that this is a very unique phenotype, and being able to determine that you have abnormal vasculature in hemophilia is just already a really big step towards maybe solving this problem of aberrant vasculature. So next, I was given that I couldn't really find anything from other joint diseases like rheumatoid or osteoarthritis, I decided to focus on the reason for the abnormal vasculature. So as I mentioned before, hemophilia results from factor VIII deficiency, and factor VIII is a clotting factor. And so I had one of like two hypotheses. First, I thought, okay, maybe the factor VIII deficiency is also causing the abnormal vessels. Maybe it's just one big pathway that's causing both hemophilia and abnormal vessels. The other hypothesis is maybe it's the joint bleeds itself. So you'll have a joint bleed, and then when the vessels regrow, because of the extent of the joint bleed and the hemorrhage that occurs from that, that could be what's causing this abnormal vasculature, and it could have nothing to do with factor VIII. And so in order to do that, I basically compared two different models. So one of them was a factor VIII knockout mouse, which means that factor VIII was completely removed. And then I had a factor VIII inhibitor mouse. So this was a mouse that was injected with a factor VIII antibody, an anti-factor VIII antibody. And I also injected it with anti-TAFI, which is another protein that someone in the lab was looking at and had determined that it was also very important in the formation of, or in the development of HA. And so I wanted to compare these two. So by looking at only knockouts, we have a mouse that is completely devoid of factor VIII. And then we have an inhibitor model where we can simulate the same injury that you see in a factor VIII mouse, but then when the vasculature starts to recover, when the vessels form again, you still have factor VIII. And so I'm able to determine if it's factor VIII that's causing abnormal vasculature. So I induced an injury, and this was done by subpatellar puncture. So basically you take a needle and you stab the knee of the mouse. And this induces a joint bleed that's similar to what you see in humans. And I measured hematocrit levels, which is basically the number of red blood cells in the blood. So 2 days after injury, you see that the knockout and wild type have the same red blood vessels, and the injured and knockout and inhibitor have the same have the same red blood cell count. So that's important because it shows that prior to looking at vessels, you still have the same extent of injury. So when we compare vessel formation, it has nothing to do with the extent of injury, like that was a control. So then I looked at alpha SMA vessel count, and the results here were really interesting because, as you can see, the knockout vessel count is significantly greater than baseline and the inhibitor models, both 2 weeks and 4 weeks post-injury. So this and this is true for both of them. And with the inhibitor model, there's no significant difference between when factor VIII is inhibited and the baseline. So it is definitely the deficiency of factor VIII that is causing abnormal vessels, and not the injury itself that's causing abnormal vessels. And so this is important because from this, we like knowing the protein that is causing these abnormal vessels, it's definitely a step in the right direction towards potentially treating them. So that was my next step. I wanted to know what angiogenic markers might be expressed in these factor VIII knockout models that can serve as treatment targets. And I used factor VIII knockout models and not the inhibitor models, since in the past experiment, I determined that it was factor VIII deficiency. And angiogenesis is the formation of blood vessels. So being able to see what proteins are causing vessels to be formed is really important, because this will show like what proteins are being involved in abnormal vessel formation. I think someone maybe raised their hand. I could be wrong, but.
Yeah, let's keep our questions till the end and then we'll call on you.
Okay, perfect. So for this, I originally tried to do normal immunofluorescent staining, which I've described before. And how that works is, for joints, you will take the joint tissue, you'll decalcify it because it has bone, and then you'll fix it in paraffin, which is a wax. And then you'll take sections of the wax and then stain it with the primary and then secondary antibody. And so when I used this paraffin method, I had used it to stain for CD31 and alpha SMA before, which is what you saw in some of my other like staining figures that I had put in the presentation. But when I tried to stain for angiogenic markers, I didn't find anything. And that was really surprising, because you definitely expect to see angiogenic markers with the type of vasculature that you have. And after doing a lot more research, I realized that the steps of chemical fixation and decalcification actually reduces the ability of these antibodies to bind to the tissue. And because of that, it's really difficult to stain using paraffin. And so I was like, okay, back to optimization of procedures. I need to figure out a way to stain this tissue that removes the decalcification step and removes the chemical fixation step. And so there's something called cryopreservation, which is where you basically snap freeze the joint in a block of ice. And so from that, you're able to pretty much still have fresh tissue, and you're removing the chemical fixation step. And in order to prevent to not have to decalcify the tissue either, I ended up using a diamond-coated blade that was basically just able to cut through the bone itself and create pretty decent sections. And I would have to switch out the blade pretty often, but I was able to optimize a cryopreservation technique using those two factors, and found that it was able to stain for a variety of angiogenic markers. And one of the main ones I saw was VEGF-R2, which is vascular endothelial growth factor receptor, and neuropilin-1 and neuropilin-2. And so given this information, I wanted to ask the question, So let's say I inhibit one of these angiogenic markers. What effect is that going to have on the vasculature? And so I chose VEGF simply due to like the amount of VEGF that was shown through staining. And I found something really interesting. First, I, again, measured the hematocrit levels 2 weeks post-injury, found that they were the same with the knockouts 2 weeks post-injury and with anti-VEGF. And when I looked at the alpha estimated blood vessel count, I found, wow, look, the anti-VEGF treatment goes back to baseline 2 weeks post-injury. And that was really cool because it showed that while there's a significant increase in the knockout mice 2 weeks after injury, which is expected, with VEGF, you go back to normal. But then here, 4 weeks post-injury, there's no difference between the knockout and the knockout with treatment. So that was both exciting because we saw that the vasculature was normalized, but it was only for a short period of time. And I wanted to understand why that was. So like, why doesn't it work at week 4? And to answer this, I just read a lot of literature and found that one main problem with anti-angiogenic treatments in general is that the way that anti-angiogenic treatments work is they reduce the number of blood vessels. And so reducing the number of blood vessels also reduces the number of blood flow and therefore oxygen that's being given to the joints. And sometimes the body makes up for this lack of oxygen by producing more blood vessels. So you have this like counteractive effect where you're trying to reduce the blood vessels, but then it's way too much and then the body produces even more. And so I wanted to figure out, okay, is this what was happening with my experiment? Was there a lack of blood vessels? And so I decided to stay in for hypoxia. Hypoxia is lack of oxygen. And I found that this is the baseline, and 2 weeks without treatment was the same as the baseline. But then with the VEGF treatment, we see increased signal for hypoxia. So what had happened, I think, is that the VEGF treatment was a bit too potent, decreased the blood vessels way too much, and caused decreased oxygen. And this is definitely something that can be accounted for. You can always do things like decrease dosages, use a less, potent antibody, and things like that. So this is just another figure where I quantified what you saw earlier. So this is just a quantification from image A, and you can see the anti-VEGF has greater density of red signal of hypoxia compared to no treatment. So before I go into major findings, I just wanted to elaborate really quickly on the hypoxia and like a little bit of like next steps that can be done from this project. So yes, the anti-VEGF treatment was too potent. So I think something that would be interesting would be to maybe look at the neuropilins. So the reason I chose VEGF was because of how much signal I found when I was doing the immunohistochemistry staining, but that might have actually been a bad thing because now decreasing this major angiogenic marker can hurt the blood vessels a little bit too much. So maybe using something that's a little bit less extreme, like one of the neuropilins, would be an effective solution. Also, another thing that you could do is maybe just like do different dosage experiments and decrease the dosage and find that, point where it will work and decrease the blood vessels, but not induce hypoxia. And so some major findings from my project. So I first established that there are vascular changes that are associated with hemophilia. And I found and established that these abnormal vessels are specific to hemophilia as compared to other joint diseases like rheumatoid or osteoarthritis. I then went on to identify multiple angiogenic markers that were expressed in the factor VIII knockout models, and these could serve as treatment targets. Then I decided to choose one of these angiogenic markers, anti-VEGF, and tested the efficacy of that and found that it significantly reduces vascular formation 2 weeks post-injury. Finally, I determined that it's factor VIII deficiency and not joint bleeding that is the result of aberrant vasculature. And this finding can, lead to a completely new project about factor VIII and the factor VIII pathways, and more like mechanistically what is causing this abnormal vasculature. And also just some imaging techniques. So first, the development and optimization of the micro-CT-based imaging technique for 3D visualization of blood vessels, which has a lot of different applications. And second, a method for immunofluorescent staining of joints, again, has applications beyond this project, which this method can now be used to do immunofluorescent staining of any tissue that contains bone. And I also just wanted to, emphasize that I was working at the Scripps Research Institute. I had a lot of mentorship from my PI and a postdoc that I was working with. I did a lot of the micro-CT scans at UCSD, and a lot of, got a lot of help from that. Portal, and I was able to get an anti-VEGF antibody from a company that generously donated it. And yeah, that's my project, and I'd love to answer any questions that you guys have.
Thank you so much, Ruchi. That was a very insightful presentation, especially I think you did a really awesome job of taking such a complex topic and a complex project and breaking it down so that you can see every single interesting finding that you found. So I really enjoyed that.
Thank you.
So if anybody has any questions, you can either raise your hand or put them in the chat. Maybe people are typing, so I can ask you one question real quick. So you did this in high school, correct?
Yes.
Yeah, so that is super impressive. Could you walk us through a timeline of, what kind of hours you spent working on this each week, and kind of the timeline of how much you worked in the summer over, if you were doing it over school and things like that?
Yeah, of course. So I started working in the lab on my 16th birthday, actually, just like immediately went in as soon as I turned 16. And so that was beginning of summer into my junior year. So June after my sophomore year, I started working in this lab. And I pretty much stayed in this lab until COVID started end of senior year. So I was working on this project for almost like 2 years, basically. Which kind of explains like the extent of the research, right? Because it takes time. And over the summer, I worked completely full-time, 40 hours a week. And over the school year, I worked around 20 to 30 hours a week, mostly on weekends was when I was working. And then, winter break and Thanksgiving break, I was able to go back to the full-time working again. And yeah, I did definitely work on this throughout the school year. I feel like it was a difficult project to complete in a short period of time, especially because a lot of the, optimization, it's not something that can really be rushed, or I can't really predict the timeline for when I'll be able to figure that out. So the first summer, like the first 3 months I was working in this lab, I was just focusing on figuring out a way to image the vessels. So yeah, it takes time, and research takes a lot of time. And that was to the point where I was also working, 40 hours a week during the summer for like 2.5 months. It took just for that very, very first step. So yeah.
Wow, that takes a lot of dedication. Yeah. We have a question that says, what inspired you to start with this research?
Yeah, so I'm actually I was in general too, just really interested in blood disorders and blood research in general, just based on family history. And so I knew I wanted to work in a blood lab. And so I actually reached out to 2 labs like a month before I started. And turns out the labs are actually a joint lab. Like they work in the same lab and they're 2 different PIs. So it was really awkward going in for my second interview and then realizing it was the same lab. But I ended up just working in that lab. It ended up working out because they were the same one. I didn't really have to choose. And I started this research mostly based on that past paper that was published, which I had mentioned about this abnormal vasculature. And I think it was really interesting to me and kind of shocking, like the lack of technology that was available just for imaging. So it was just something I really wanted to work on. And it ended up, having like so many more applications and steps that came as a result of that. The project definitely wasn't something that I started out knowing exactly what I was going to do from the start. I really just started out saying my project was going to be developing this imaging technique, but based on the findings from that, it really just grew and grew and became like the presentation I just gave.
All right. One person asks, what program do you usually use? And if
So
you need programming in general.
no, I don't lead program. I used ImageJ analysis, which you saw a lot. So that was to determine like any of the immunofluorescent staining data. So that I did myself. The micro CT scanning was the analysis was done through MATLAB, which I actually partnered with a lab from UCSD. And so a postgrad student coded the MATLAB images. Yeah.
All right. We have a student who says, I'm in the process of writing a scientific article for my own research. Congratulations. I was wondering, do you have any tips for writing one?
Yeah, I mean, I've never personally written a scientific article before. I have written like research papers. I think the best thing to do is to find someone who can edit and look over it. I think that like, especially when you're the primary author of a paper at our age, it's really difficult to really match that level of like difficulty, I guess, that you normally see in scientific literature, especially just like getting into the habit of writing scientifically is very different. So I think find someone that will be able to consistently read over your work. What I would do for my work is I found someone and I would set up like monthly meetings with them. So I would work on my paper and then I'd send them the paper. We'd go through it. She'd make edits and then send it back. And then a month later, I'd do that. This went on for like around 2 years before it was finally actually published. And I think like the best thing you can do is just find someone that will edit it for you. It's going to be a very slow process though. I think for me, it just happened to be very slow because it required a lot of edits. And I think it's hard to match like a PhD level paper when you're a high school student. And I think just knowing that and keeping that in mind would also be good.
Yeah, and I think our next question, you briefly touched on this, but what extent would you say mentors helped in your project, and can you do a project without them?
Yeah, so I'd say in the beginning, the part that my mentor helped most with was learning how to do techniques. So I had never worked with mice before this project. So that was something I had to learn. And a lot of these techniques is not something that a lot of people just know going in. And I think until you're at the level where you are able to formulate a project where you know how to do everything and you know how to do every single experimental protocol, I think it would be helpful to have a mentor. And also just a mentor is a great person to learn from. And the point of doing research in high school is to like learn these new techniques. So it would be great to do a project where maybe you don't know how to do it. So that's a learning opportunity for you. It's definitely possible to do a project without a mentor. In the lab that I'm currently working in, I'm actually the only researcher in the lab. So there's no one to ask for help. So I just can't have a mentor in my current research project. And it's definitely something that I can do, but because of my experience with research in the past, that is what has allowed me to do a project without a mentor. I don't think I could have started it and started research in general without any guidance.
Right. Valeria asks, how did you prepare for this research, and how did you get all the knowledge you needed to start your research?
A lot of paper reading. The best thing you can do is start with the papers from the lab that you're working in, because chances are the project you're working on isn't something that's completely new and has like the disease that you're going to be studying is something that the lab has probably been researching for a while. And so reading through the papers, reading what's out there, especially reading the methods and materials section, because those are likely the experimental procedures that you're going to be working on as well. That's really important. And just following up with just general findings in the field. So like looking at hemophilia A and vasculature, it was hard for me because the only papers that had come out on the topic were ones that came out from my lab. And so I think for me, the amount of like data in general was very limited. So it wasn't too much effort, I guess, or it didn't take too long to read through all the research out there. But I think the most research I had to do was when I was doing like optimization, or like, for example, when I didn't know why after week 4, the anti-VEGF treatment wasn't working. Like I always just went back to reading papers, seeing like, oh, why do antiangiogenic treatments not work? And so, yeah, I think, sorry, I hope I answered the question. Yeah, I guess, yeah, a lot of my knowledge came from reading papers. You can also ask my mentor a lot what papers to read. So like, if I don't know something, you can always ask someone like, hey, can you suggest a paper that I can read for that? And you don't have to do everything on your own.
Right. And I think that's a really important point, is that to utilize your connections and, you know, help other people can also like help you find out what you don't know. So there's like things you know, there's things you don't know, and then there's things you don't know that you don't know. So mentors can be really helpful with that. I just wanted to add that in real quick. Okay, over the past 2 years, that's a long time to do research. How did you keep moving on?
Yeah, I think like my research kind of lent itself to follow up research. And even when it didn't, like, for example, I realized, oh, you don't see this this phenotype in any other joint disease. Like not every experiment you do is going to lend to a future experiment. And I still think that it's important to include, like I still showed this experiment in my presentation because it did lead to findings. But I think like continuing your research just has to do with like keep asking questions. So like when something happens, when you get a result, you can ask why. Like, okay, I see abnormal vasculature. Why? Is it because of factor VIII? Is it because of the joint bleeding? Do an experiment for that. You can see it's because of factor VIII, or you can see that there's increases in vasculature. You can say, oh, how am I supposed to, how am I supposed to account for this? Like what proteins are being expressed? And when you find that, you can say, like, what antibodies can I use to prevent these proteins? Is that helpful? And if there are any useful results for that, it's like, why did that happen? And it's just like, I don't know if you guys could tell, but like just like constantly just asking questions, asking why you got a certain result. How can I proceed? Like knowing this information, what can I do now in the future to further progress my project? I personally think that this project could have lasted over 2 years if it weren't for being kicked out of the lab from COVID and like, leaving San Diego. But definitely like this project could have progressed, like I mentioned, from different dosage experiments of VEGF, different looking at neuropilins, eventually, looking at clinical trial models. Like it definitely could have progressed a very long way just through asking questions.
Yeah. And to be able to work inside a lab, what did you have to do? And what kind of ways did you, get yourself in the door?
Yeah, for sure. Okay, so this current lab I worked at was not my first lab. I worked at Stanford Burnham when I was 15, the year before. And that was basically really difficult to get that position. I ended up emailing around 72 labs. I don't remember. It was a lot. Over 70 labs. And one of them said yes. And I obviously did not choose my project. I was strictly a learning experience. And I think it's important to kind of have that expectation for yourself that the first lab I work in, I might not be leading my own project, right? Like you might be just learning and getting used to the lab environment. And that's also really important. And I think that experience is what really helped me get the second opportunity, where I only emailed these 2 labs and was able to get offers from both PIs for that. And so I think having past experience was definitely helpful, and showing that you are very valuable to the lab and that you can actually contribute. I think, being more helpful than maybe like more of a burden, because not everyone always has time to like teach you things. So I guess showing that you have a lot of initiative, you have what it takes to learn, you're a fast learner, you'll put in the time, you'll put in the effort, just proving those things to your mentor and to your PI is very important.
Thank you. Someone else asks, do you think this research influenced your decision for a major?
Oh, for sure. Yeah, I think I was interested in bio like before I started this project, but I think like specifically molecular cellular bio, which is my current major, it's really interesting. And through this project, I think I was able to like look more at the applications rather than just textbook learning of what is molecular and cellular biology. And I think having this experience now, taking classes in college, I really am able to see like a lot of applications of the things that we're learning, and it makes me a lot more interested in the classes that I'm taking. So it definitely didn't influence my major, and there are like 3 different biology sub majors, I think, here. So I think it definitely influenced me choosing molecular and cellular.
All right. What science fair competitions did you do?
Yeah, so I actually entered the science fair in 8th grade, was my first time. I did an interesting like overextending it. I did a project on looking at bacteria in sushi, and I wanted to see if it was pathogenic or not, because we were eating raw food and I thought that was weird. And that was the first time I entered the science fair, and I loved it. And I did it again in 10th and 11th grade. So this is the Greater San Diego Science and Engineering Fair. And I eventually was on the science fair advisory board for that and helped run the fair every year, which was also really fun. And so, yeah, I did that. And then my junior year, I qualified to the state science fair as well, where I placed first, which qualified me to the international ISEF International Science and Engineering Fair. And I also participated in the Junior Science and Humanities Symposium in my junior and senior years, but the national competition was canceled the senior year, so I didn't participate in that one.
All right. And yeah, and to add on, if you're not from Southern California, I think every state has some local science fair. You just need to check that it goes to the state science fair, and eventually you may be able to go to ISEF if your project is really cool, like Ruchi's. Let's see. More of a
Also, I have
personal.
something to add. I also entered in the GSDSEF, and it was a great experience. And I'm also in the student leadership board.
Yeah, nice. Right. More of a personal question. What other activities did you do in high school?
Yeah, so I think, okay, most of my high school was research. I think just as I explained, given the amount of time research took, I didn't have a lot of time for other stuff. But like I said, I was on the student advisory board. I helped lead workshops where I was basically teaching people how to lead science fair projects. I started 2 programs in other schools that were very similar, where I was basically doing a more science fair program where they eventually submitted to fairs, or I hosted a fair within the school. I was also, unrelated to science, I was on the debate team. I did Lincoln, Douglas, and public forum debate. I was a competitive debater in that. And I also did like hospital internships. I shadowed some people, like some doctors. I was also a teen ambassador for this human trafficking organization. Yeah, a lot of it's not like very STEM related, mostly because I have other interests, but yeah, I think I was able to find a good balance between, you know, debate and research.
Wow, an impressive resume. Another participant asks, where did you publish your research paper, if you published at all?
Yeah, so one of my papers was published in the Journal of Clinical Investigation. Another first author paper was published in Cancers. I had some like just conference publications in like various national conferences as well.
All right, I think you might have touched on this before, but how did you outreach all these labs? And do you have any tips on how to reach out and find these types of opportunities?
Yeah, I think the most important thing in your email is to keep it short and persuasive and also very specific to the lab that you want to work in. So explain why you want to work in that lab. And of course, attach your resume to the email. A lot of it's just cold emailing and following up. So like even if someone doesn't respond within a week, just be like, hi, I wanted to follow up on this last email, and chances are they'll respond to the second one, even if it's a no. It shows persistence, which I think is really important. And I think another tip I have, I guess, is if it is your first time working in a lab, to not be quite as picky with what type of lab you're researching in or like what your project might be, because it's going to be really difficult from what I realized to like find a project that you really like until maybe your second or third lab, you know, when you have a little bit more choice in the lab you're working in.
Right, and then I also wanted to add that over the summer, a lot of universities include or conduct their own camps, like research camps. You could attend those. Usually you have to pay for those, but and like finding your own lab and showing that initiative, that can get you closer just to the principal researcher, and they can be more involved. Whereas these camps, they're more tailored and have a more rigid structure, you usually have less time. But yeah, that is really good advice, Ruchi. Do you have any advice for incoming high school freshmen who want to get into research?
Yeah, I'd say, I mean, very similarly, like be very flexible with just using your first opportunity as more of a learning experience than like an actual leading a project. And just be open to like, you know, learning in that lab environment. I think another kind of thing is just like talk to people and like read other people's research. I honestly didn't think I would be able to lead my own research project until you know, I was in 8th grade and I went to the GSD SEF and I saw the high school project, and I was honestly just like blown away by like how complex these projects seemed, and the boards were so tall and like the figures looked so complicated, and I was like, wow, I can't believe like people only a year or two older than me are doing this. And I think like a lot of it is just like having a good mentor to learn from. And something I also want to emphasize was like, I think personally in the end, having a good mentor is more important than the type of research you're doing. Like if you're choosing between two labs and you like one project better because you're more interested in the disease, but you have a mentor that's more willing to like put in the time, I honestly would say like go with the mentor. Just find a good mentor who's gonna guide you and who's really gonna put your best interests first, because if they're, you know, truly interested in giving you an opportunity to learn, then you'll also get the most out of it.
That's great advice. Okay, this one is sort of related to things you were already talking about. So that when you were actually emailing the labs, did you already have an idea of what you wanted to research in your mind, or were you just like, hey, I want to do research?
Yeah, for sure. I think so when I was first emailing for my first lab, I originally had an idea, but they all said no. So I had to like let go of that and you know, just email labs in general. So I did have an idea. It did end up, you know, I didn't end up actually doing research on it. But I got a lot of experience from that lab. And then the second time around, I did know what I wanted to research because the two labs that I had chosen, I had read their papers before emailing them, which is how I knew I was interested in their work. And the idea of just like spontaneous recurrent bleeding was something that I had, you know, seen people in my family struggle with, and it was something that I knew I wanted to research. And so I did know the second time, I guess I knew both times, but the second time around, I was able to actually research what I wanted to.
All right, people are asking for where they can find your research.
Yeah, if you go on like PubMed, I guess, and type in my name, you should find papers. I mean, it's just like a online research database that has all papers in it. So you can search my name up. You can also find, I guess, abstract publications won't be on there, but I think if you search my name up on Google, you'll find the abstract publications as well. If you just, you know, search my name and write like hemophilia or something after. Yeah. I think they should also be on my LinkedIn if you just want like a list of the publications and like links to them. I guess you could find it on there too.
Okay, we have a bit of an interesting question. Is mentors stealing the idea of a student and using it for their own work a thing?
Yes. I don't think it would necessarily be called like stealing ideas because in the end, I think my project was a part of an even bigger project that was going on in the lab. And so, you know, my work might be included in a lot of these papers. My like direct figures that I showed you in this presentation have been included in papers. And I think that because I didn't specifically write out and type out the manuscript for this entire paper, it makes sense that I'm not like the primary author. It's the person that, you know, did this whole research project. But I think you should get credit for the research. Like if they are using your figures, if they are using your findings, you should be listed as an author. And at the bottom of the paper, like it'll tell you, you should be able to see exactly what each person's contribution was. So I wouldn't call it stealing unless they just completely just don't put your name on the paper and give you no credit in the paper at all. And honestly, like I've talked to my PI about this as well. Not something that like he was basically saying that there's a very big threshold for your name to be included on a paper. So if you think you deserve credit, and you can always just bring it up to your PI and they might just straight up tell you like, honestly, your experimentation was very valuable, but like based on just the journal's requirements for who can be listed as an author, your contribution to the research just was not enough to make the cut. And that is possible, and it's sad, but it does happen.
Also, we have a question about patents. So you said you developed the first ever visualization method for blood vessels. So do you plan to patent that? What do you think the process is? And again, you know, how does it work with your PI?
Yeah, I think it could definitely be patented. The findings are already listed in papers, so I've already been given credit for that. So I don't care too much about, you know, labeling it as this is my finding. I think, you know, having that information out there for other people to use is just as important to me. To prevent, I guess, I think getting a patent is realistic. I know people that have patented technologies that they've done in their research. A close friend of mine did that with her project. And it's definitely something that is possible. I guess it just wasn't a priority for me, given that after I did do this, my main goal was to, you know, further progress this actual project. And to me, like, working on getting a patent for my very first finding would take a lot of time out of, doing the other 4 steps of my project.
All right. I actually have a question for you. So I know you go to Yale University, which is known for its research. Are you conducting your own research right now? And in general, what would that be? And what's the process of like finding a mentor to work on your research there?
Yeah, so my research is not related to this at all right now. I have a lot of interest, and I did when I was 15 when I wanted to work on blood disorders, and I'm obviously still very interested in this, but since then I've become really passionate about reproductive sciences and the reproductive medicine field, and specifically I think working on that, so that's led me to find research in the Department of OB-GYN. And so that's my current research. I really loved my PI. I talked to him, and he's just so nice and cool. And I think being the only person in the lab, it has actually been really nice because I get a lot of attention from my PI. I get daily check-ins at 5 p.m. every day. He's constantly making sure that I'm doing okay with my research. And that's honestly something that's very hard to find. Normally, the person you're working under will be like a grad student or a postdoc. Being able to work directly under your PI as a undergrad or high school student is something that's less heard of, I guess. And so that was something that I was really looking for. So one that more like independent research. I had experienced that in high school, and I personally like having my own timeline, being able to determine what I'm researching. So finding someone who trusted me enough to do that on my own, and also finding someone that was doing research in the field that I'm interested in as well.
All right. And with that concludes our webinar with Ruchi. If you're on YouTube, you can click the links in the description. Those will have links to Ruchi's info, and in addition to all the info that Sarah mentioned at the beginning that's related to Helix in general. So with that, thank you so much, Ruchi, for such an engaging presentation and answering and taking so much time off of your Saturday to work with us. Thank you so much.
Thank you so much.
Thank you, Ruchi. I really appreciated you speaking here, and I learned a lot, and I'm sure that our audience also learned a lot. So once again, thank you so much.
Yeah, thank you. Have a great weekend.